In this video, we'll be looking at a case of steroids gone wrong. Actually, we'll be looking at the difference between antibodies and antibiotics. This is a common confusion. When I was also doing the IB, this was super confusing to me. I couldn't understand the difference between them. But in this video, I'll make it super clear and it'll be super easy to know what their difference is and everything you need to know about them. So let's get into it. So first, I'm going to explain what antibodies are. You probably have learned about antibodies if you've learned a little bit about the immune system, okay? So let's start. Antibodies, so let's start here. We know pathogens invade our body, right? When pathogens invade our body, we have, so these two are pathogens, bad things entering our body. We have these cells called lymphocytes. These lymphocytes can bind with their receptor to the pathogen's antigen, okay? This antigen is this V-like shaped thing that tells your body that it's a bad thing. So they know, ooh, this is a bad thing, we gotta bind to it and kill it. So as soon as this lymphocyte binds this antigen, it starts to differentiate or become two different types of cells, either a memory cell or a plasma cell. Plasma cells are going to secrete antibodies, okay? And these antibodies are like weapons made to defeat and target this specific pathogen. So it looks like this. It secretes a whole lot of antibodies, and so eventually when these antibodies encounter again this pathogen, like this, so say it finds this pathogen is swimming around, and eventually its antibodies find this pathogen, it can kill this pathogen through various ways, okay? So here I show you three. Okay, it can make them clump together, it can destroy the cell itself just by ultimately obliterating it, or it can bind to, or the antibody can bind to the antigen and just destroy it through any other mechanism, okay? You don't have to worry about this for now, but just know antibodies are things naturally made by our bodies, by our immune cells, specifically our plasma cells. They are made to destroy pathogens that enter our body in a specific way. So let's look at the key points. Number one, they are made naturally, and by naturally I mean by our immune system, against pathogens that invade our body. That's point one. There's going to be various things that you need to know. So point two is antibodies can kill various types of pathogens. So what I mean by this is this antibody, okay? It can kill a virus, it can kill a kill a bacteria, it can kill a parasite, it can kill many types of pathogens. Okay, so it's not like antibodies can only kill bacteria. No, they can kill various types of pathogens. Bear this in mind. Okay, antibodies are specific, meant to kill a specific pathogen. So these 2 points might confuse you a bit. I just said that they can kill various types of pathogens, but then here I say they're meant to kill a specific pathogen. What does this even mean? So when I say various types of pathogens, I mean it can kill viruses, bacteria, parasites, all these different types. Like they're not restricted to only killing bacteria or viruses. They can kill all of them. And what I mean by killing a specific pathogen is that when this lymphocyte binds this specific pathogen, this antibody produced, these antibodies, will only be able to kill the pathogen that it binds to originally. So this antibody will not be able to kill this pathogen because this pathogen is not the same pathogen that the lymphocyte originally bind to, okay? So that's what I meant by they kill a specific pathogen. So this antibody will only be able to kill the original pathogen bound to, okay? So this one. But say originally it bound to this pathogen and made antibodies. Now those antibodies can't kill this pathogen because those antibodies are specific to the original pathogen that the lymphocyte bound to. So in this case, it will only be able to kill this kind of cell, this kind of pathogen, okay? Okay, last important point. Antibodies bind to antigen and kill through various mechanisms. This one we already discussed. These antibodies will bind to the antigen, so any of these surface molecules or these surface structures, like that right there, or if we look at this one, the V shape, it binds to these antigens and eliminates this pathogen through various mechanisms, okay? So this is all you need to know about what antibodies are, okay? So next I will introduce what antibiotics are, and then I have a small table summarizing and comparing their differences, okay? So stick around to see that. So now that we know what antibodies are, what are antibiotics? So antibiotics, when I think of it, I immediately think medicine, because that's what it is. Antibiotics are used when your immune system, so this whole process, isn't enough. These bacteria or these pathogens that your body is trying to fight off is too strong. These antibodies are not good enough and they can't kill the pathogens fast enough. In that case, you need antibiotics and medicines to help you do so. Okay, so for example, we know our cells, our body cells, are human cells, right? They are eukaryotic cells, meaning they have a nucleus, a lot of organelles, and all these kinds of things, right? Whereas a bacteria, a prokaryotic cell, has DNA, but they don't have a nucleus. Okay, so that's their difference, the main difference. There's many other differences, but for now, this is the only difference you need to care about. And then there's viruses, okay? So viruses are dead. They are dead things. They don't actually have any organelles. All they have is some DNA, some kind of DNA, but they have no organelles to build anything or synthesize anything. All they have is the DNA, because we know we need all of these organelles to make stuff and make the cell live, which is what the virus doesn't have. So bear in mind that a virus can't live by itself. A virus, unlike a bacteria, needs your human cell to survive. So when you're infected by a virus, that means they go into your cells and use your own cells' instruments or machinery. So all these rough endoplasmic reticulum, all this Golgi apparatus, everything, it uses all of these machinery to synthesize its own things. So it's like it goes into your cell and it's like, okay, synthesize or make me this or I'll kill you. Make me this or I'll kill you. So that's basically what a virus does. It hijacks your cells and tells them to make stuff. And all this things that it tells your cell to make is bad for you. So it could be like, okay, synthesize another one of me. Synthesize this thing that is bad for you. And like it's basically just a pure criminal. Okay. Whereas a bacteria doesn't invade your cells. They can infect your body and live outside your cells and do their own thing. Okay. Remember, bacteria can also be good at times. So let's look at the key points directly and compare with the antibodies. So antibiotics are made artificially in a lab. Okay, that's why I said it's medicine. Your doctor can give you this. Your body didn't make it. Your doctor had to give you it. So they're made in a lab or made by other organisms. So either your scientist can create this antibiotic that is going to help you destroy the bacteria, or it can or the scientists can get this antibiotic from other microorganisms. So you're going to be confused what I mean by this now. That's why I put a sun here. Later in this video, I explain exactly in detail what this means. So for now, know that antibiotics are made in a lab or by other microorganisms, so other bacteria. And essentially, the scientists will steal that thing that these other microorganisms make and then give it to a human, which will help that human defeat the bacteria. Okay, I'll get to explain this soon. So don't worry about this now. Just know that it's not made by your own body. It's made by other organisms or synthesized in a lab. That's the main difference. Okay, antibodies kill only bacteria, not viruses or other types of pathogens. So in antibodies, we know can kill anything. It can kill bacteria, viruses, parasites, anything, as long as it binds to that specific pathogen directly and makes antibodies for that specific pathogen, right? So, but in this case, antibiotics can only be used for bacteria. You cannot kill a virus like that. So let me explain what that means. Remember how I said bacteria don't invade your cells? Yes. Since bacteria don't invade your cells, when you eat medicine or take medicine, that medicine can reach the bacteria and bind to the bacteria and kill them. Okay? Or it can go into the bacteria and stop the bacteria from making stuff that allows it to survive, and that way the bacteria dies. So it can kill the bacteria basically by inhibiting it from synthesizing things that allows it to survive. But since a virus does not live outside of our cells and rather inside, then this kind of this antibiotics can't reach the virus because it can't penetrate our cells because we have eukaryotic cells. Antibiotics only work for prokaryotic cells. So since we have different type of cell, it can't enter our cells and then kill the bacteria. So essentially the virus is basically hiding from it by staying in our cells. And on top of that, even if these antibiotics could go into the virus, it's useless because the virus is already dead. There's no metabolic processes going on inside the virus. There's nothing to block because since the virus is not making anything, it's telling our cells to make stuff. If these antibiotics go into the virus, it's not going to do anything because the virus isn't doing anything in the first place. And so if you try to prevent it from doing something, trying to prevent it from surviving by making certain proteins and survival factors, then these antibiotics will be useless because the virus isn't doing that in the first place. Okay? So memorize or by this way you can understand that antibiotics are only used to kill bacteria. That's why when you're infected and you feel sick, you can't assume that you just need antibiotics because perhaps you're infected by a virus, like the flu virus. In that case, antibiotics are useless because they don't fight off viruses. But if you're infected by a bacteria and you know that because you went to the doctor, then antibiotics is probably a good idea because it will work. Okay. Next point. Antibiotics are non-specific, so can be used for more than one type of bacteria at once. Okay, so remember in this one, I mentioned that antibodies are specific, meaning when you make an antibody, it can kill only the type of bacteria or pathogen that it made the antibiotics that it made the antibodies for. So in this case, this antibody is made for let me show you. So in this case, this antibodies is made for that specific pathogen. It can't kill this one, right? That's what I meant by specific. Now, in the antibiotics case, it's non-specific. So when you administer, say for some reason you're infected by two bacteria or two kinds of pathogens, so this one and this one, and then you decide to take these antibiotics. So these are two bacteria. Remember, it can't kill viruses, only bacteria. So say you're infected by two viruses and you take antibiotics. These antibiotics can simultaneously kill this bacteria and that bacteria. Okay, it can go into both of them. Antibiotics are not specific because if they were specific, it would mean this antibiotic that you took the other day would only be able to kill this pathogen and not this one. Antibiotics can generally kill many bacteria. Okay, so any type of antibiotic you take will most likely be able to kill any kind of bacteria. They're very diverse. They're not specific, which is pretty nice. Otherwise, every time you had a bacterial infection, the doctors have to make a specific antibiotic that is specific to that pathogen. So it's pretty nice that they're not specific. That means if you're infected by a bacteria, you can probably just take any antibiotic and it will work. Okay. Last one here. Antibiotics don't bind antigens. Okay, I'll fix this. They destroy cell wall and prevent cellular functions, metabolism of bacteria. So like I said with the antibodies, they bind to the antigen of the pathogen and then destroy them through various mechanisms, right? Well, antibiotics don't do that. They won't go and bind to this antigen and then destroy it. They go into the specific bacteria and then prevent the bacteria from synthesizing important proteins that allow it to survive. So that's basically what they do. They don't bind to the antigens and kill them that way. They go into the cell, prevent the cell from making important things that it needs to survive, and that's why they end up dying. Okay? It's basically like imagine a human. If you told a human they can't eat, that's basically what this is happening. These antibiotics are essentially going into a bacteria and preventing it from eating and surviving, doing everyday necessary things to survive. Okay? Okay. That's what I meant by metabolism, because metabolism is any process that allows a cell to survive. Okay? Now, here I have a nice summary. So here I compare. So for example, in antibodies, I will write specific thing and contrast it or compare it to how it works in antibiotics. Okay, so hopefully this is useful for you, this summary. Now, for the last section of this video, I'm going to be talking about how they discovered the first antibiotic, what the first antibiotic is, and what I meant by other microorganisms synthesize them. Because remember, I mentioned this point here and I wasn't so clear on it, so I'm going to try and talk about that now. This next section is very important also for IB, so make sure you stick around to watch it because it's commonly asked on multiple choice questions. So here I show you an image, and right now you're going to be confused as hell, because I was confused as hell when I saw this image. I was like, what? Penicillium colony, staphylococci undergoing lysis? I was like, I have no idea what's going on in this. So that's my job here, to help you understand that. So let's see. This right here, this circular thing, okay, is this. Okay? And what this round kind of plate thing is what scientists use to grow bacteria in. You can see this red kind of liquid or gel. This gel contains all the necessary nutrients or foods that a bacteria will need to survive. Because if you try and grow a bacteria on an empty plate somewhere, it won't grow because there's no nutrients. So they put this sort of nutrients in this plate so that any bacteria that is on there will grow and survive and eventually become bigger and bigger and bigger. Okay? So that's what this circle is. Okay, just so you know. So it's called a petri dish. Now, this image, these words are confusing, okay? So I'm going to explain what's going on in this picture step by step instead of just explaining the image, okay? So let's get to it. So basically, there was a scientist, I'm not going to mention their name, and what they did was they had a petri dish, just like this. And on this petri dish, there was this white things growing. And these white things growing were groups of cells, okay? And these cells were bacteria cells. So, for example, one circle here represents probably like 1,000 bacterias, okay? So they're growing in groups, like little families everywhere. Here's a family, there's a family, okay? They're growing and getting bigger and bigger and bigger because there's nutrients that allows them to grow, okay? Now, in this specific experiment, the name of this bacteria was called staphylococci, okay? Staphylococci bacteria. And let me help you memorize this name. So staph means grape. Okay? Grape. That's what staph means. And cocci means balls. So it's like this bacteria looks like grapes because staph for grapes and cocci means balls, which is the grape balls or the grapefruit. Okay? So when you look on a microscope at staphylococci, because right now it's a big circle, and this big circle doesn't represent one bacteria, it represents thousands. So how does one specific bacteria look like? So that's what I'm going to show you here. It looks like grapes. That's why it's called staphylococci. So let me show you. If you zoom in on a microscope, this is what you see. You see a whole bunch of cocci, or balls, stuck together like it is in a grape. Okay, that's what this bacteria is. It's a grape-like bacteria because it's a bunch of balls or circles sticking together. Okay, that's what this is. So that's what this name means. So he was basically growing all of this staphylococci on this petri dish. And they were getting bigger and bigger and bigger until one day he realized something. He realized that something was growing on one end of this petri dish. So in this specific picture. So this, by the way, this picture is the picture he took in 1928. So I'm just trying to make it clearer. So he noticed in this top corner, so same as here, something was growing. And what was growing was something called penicillium. Okay? And penicillium is a fungi. Remember, you have to know about different kinds of pathogens. And fungi is one of them. Just like bacteria and virus is one of them, fungi is one. So there was something growing here, fungi. It was growing and getting bigger and bigger. And then he noticed something. He noticed after a while of this fungi being there that all these bacteria close to it were dying. Okay? And so he couldn't understand why. And what he was thinking was the only reason that these bacteria could be dying around this specific fungi growing is because this fungi is making something that is killing the bacteria. Okay, and that's what he was thinking, and it ended up being that this specific thing that's killing all these bacteria was penicillin. So these fungi were making a drug called penicillin by themselves and cause all these bacteria to die around them. Okay, so that's what I meant when I said other microorganisms can make it, because essentially after the scientists discovered that these fungi were making a drug killing bacteria, they were like, holy crap, how about if we can find a way to extract that magic thing that this fungi is making, we could use that to treat other people who are infected by bacteria, because it seems that this thing is killing the bacteria. So if a human is infected and they can extract this kind of magic drug and inject it into humans, then the humans could survive, right? That was their logic. Okay, so that was the discovery of penicillin, and named after the fungi Penicillium that makes it. Okay, that's what penicillin is. So I'm sure you know what penicillin, you've heard of penicillin before. So that's how it was discovered. So humans did not make penicillin. Fungi, a specific fungi by the name of Penicillium made it, and humans just extracted it. And basically, after they extracted it, they looked at its structure, and now they're able to synthesize it and make more of it. And as they make more of it, now when people get sick with bacteria, they can give this penicillin to people that will fight bacteria just like it did here and allow people to survive. So I hope so that's pretty much it's a bit less clear in this image. You can see all the colonies. So colony is just a group of bacteria or a group of cells. So you can see all the zones around the fungi, there's less cells because they're being killed. So lysis just means to be killed. And then all the ones far away were surviving. Okay, I was meant to use this. Just penicillin, fungi, staphylococci, explained already with the grapes. Okay. Now in the modern experiment, they redid it recently, and basically they made lines like this, and these lines are just small bacteria. If you zoom in to these lines, it will just be small bacteria, and you can notice all the bacteria right around the colony. That fungi colony again is dead. You can notice, right? There's a small patch here where the bacteria are not surviving, even though all the ones far away, so all of these, are surviving. So that's just a modern day image. It's the same thing as what he did here, just a modern day version. So if you understand this one, you understand this one. It's the same thing. The only difference was in this one they made small colonies of bacteria, whereas in this one they made rows of bacteria. So they made one row, another row, another row, another row, another row, instead of specific colonies. And it shows the results a bit better. Now, there are 3 people that you need to know about in the IB, called Alexander Fleming, Ernst Stein, and Howard Florey. Alexander Fleming is the guy who noticed this phenomenon. He's the guy who was growing the bacteria, and then accidentally these fungi were starting to grow, and it caused this kind of shape. And so he was the one who noticed that the fungi were making something that was killing the bacteria. He was the one who noticed that. And then this guy, Ernst Chain, he was the one who isolated penicillin. So he isolated this specific substance in here that was produced by the fungi that were killing the bacteria. And he gave it the name penicillin after the Penicillium fungi who makes it. And then the last guy, Howard Florey, he's the guy who used this penicillin. He used the penicillin that was isolated. Isolate just means to extract from. So he used the penicillin that was extracted from here, and he gave it to different people to see if it works. So he gave it to some sick people to see if it heals them. Okay, and that's what I'm going to show here. So essentially, before he tested on humans, he had to test on animals, right? Because animals animals behave more similarly to humans. So if it works on an animal, it is most likely to work on a human. So this is what he did. He took 8 mice, okay? Then he infected these mice with a bacteria, a deadly one. Let me show you. A deadly one by the name of, I spelled it wrong, Strepto. You don't have to know that name. Just know that He injected them with a deadly bacteria called streptococci, a lethal dose, meaning it can kill them. So basically, at this point, all of the 8 mice had this bacteria. Then he gave penicillin, which we know is this drug that they extracted that kills bacteria, the first ever antibiotic. So this is an antibiotic, by the way. So he gave this drug, this antibiotic that was discovered, to 4 mice. So let's say the first 4, 1, 2, 3, 4, and he didn't give it to the other 4. So what do you expect happened? So let me show this more clearly. Okay. So these 4 had the drug, these didn't. Which one survived, which one didn't? So the 4 that had it survived. And then the 4 that didn't have it died. Okay, so this is how they this is what in this experiment they essentially proved that penicillin is an antibiotic and that it helps to kill bacteria when organisms that are infected by it. It can basically help organisms that are infected by bacteria by killing that bacteria. So hopefully this made sense. I hope these diagrams. I hope that you can remember the people and the main differences between antibodies and antibiotics, and the process of how the first antibiotic, penicillin, was discovered. So other than being discovered like this, because ever since this happened, many antibiotics were discovered from organisms that produced them, and they were extracted just like penicillin, and they're used now every day. But there are also other kinds of antibiotics that are synthesized in a lab, so not discovered by like this. They're made by scientists and things like that. So